Hi friends, welcome to Science Teaching Academy. Today, this is the topic Cell Division Lecture 2. It's a follow-up lecture of Cell Division Lecture 1. So friends, before seeing this, I recommend you to see the Cell Division Lecture 1 too. I shortly summarize what I have covered in Cell Division Lecture 1. I have talked about what is cell cycle with a funny story and how cell division occurs and what is the significance, and started with interphase, then prophase and metaphase. We are well aware of it. Prophase is where the nuclear membrane disappears and nuclear membrane chromosomes condense, and in the pre-metaphase nuclear membrane disappears, and after that the spindle fibers is just pulling out the chromosomes to their disrespective poles, means their opposite poles. That's what we are going to see in this lecture series. We are going to continue the story of the two sisters which we seen in the last lecture. Guys, it's very simple. I have relating the two chromatids as sisters and the spindle fibers to their respective boyfriends. Oh, it's gone to the story, back to the story. Okay, fine. We have seen what happens in prophase, pre-metaphase and metaphase, and then we move on to anaphase. Again, I will repeat, guys, please follow Cell Division Lecture 1 available in YouTube, the same lecture series in the name of Science Teaching Academy, to know about these three phases well, and come again to this lecture video. Fine. Anaphase. In anaphase, the cell which form in metaphase, what happens? The cell will be in the equatorial plane or metaphase plate, pulled by spindle fibers by centrosome. The boys are trying to pull those girls towards their respective things, means to their place. Same thing happened to these sisters too. What happened? They started splitting. The two sisters splitted and they are moving towards their opposite poles. That's what life happens to them. I again repeat, I relate this spindle centrosome to boys of sister 1 and cis 1 and cis 2. That is chromatids. Okay, I will sum up biology here. Biology of this anaphase. In anaphase, what happens? Spindle pull that sister chromatids towards the opposite poles. Okay, fine. This is what happens in anaphase. And one more protein I want to mention here. These spindle fibers are attached to the protein in the centrosome. That protein name is called kinetochore, which is attached to centromere. Attached to spindle fiber. Okay, this is what I want to discuss in anaphase state. Next I will go to telophase. This is meta. Now what happens? They start pulling. Okay. Now I will say about telophase. In telophase, the chromatids come to the extreme ends of two opposite poles. The two sisters were completely separated. This is what I mean to say. And what happens? See the jealous here. The spindle fibers, the boys who pull those girls, they also disappear. What do I mean? the spindle fibers got disappeared here in the telophase state, and one more thing started reappearing. The other organelles started reappearing one by one in this telophase state, but still they are in the same home. Their home is not separated. This was the sad part of our story. Next, their home is also too separated through a process called cytokinesis. Cytokinesis, to say about cytokinesis, I will say with the plant cell. What happens in the plant cell, we all know plant cells have cell wall. So the division between their home of these two sisters happens with is an intermediate. What division happens, they all start in their home itself. That's what it happens. Cell wall form as a cell plate, which appears in the middle, and later on they move to their periphery. Finally, what happens? The two cell plates divide among themselves, and they are the precursor for two different cells. And the cell plate which form between the two cells remain as lamella in the divided cells. Okay. This is how cytokinesis appears, and the cytokinesis, at the end part of the cytokinesis, the two daughter cells have been completely divided with all its resources. So this is a successive thing, and apart from that, one more thing occurs. The two chromatids also going back to their back state of forming, revert back to their chromatin state, what their mother had initially. That is a custom in Indian society. When the daughters from their home go to their new home, means their husband's home, after marriage, we will send them equal dowry, means send all the resources along with them. That's what happened in their home of this beautiful story. Whatever the mother had, means the mother cell had in the initial stage, before prophase and metaphase, the original mother cell, from these two daughter cells form. The sisters became daughters now. Two daughter cells form. They have the same resources. What do I mean resources here is organelles, nuclear material, and everything. That's why it's called equational division. Okay. The scientific terms, if I want to say, the diploid cell, one diploid cell is divided into two diploid cells. And now we have known about what is mitosis with this simple example. Now I will say what is the significance of mitosis. Why mitosis need to occur? There are four major points to know about the significance of mitosis. Okay, mitosis significance. The first significance, whatever the genetic material we have, it should be equally distributed to all the cells, the somatic cells. Even you take a cell from my hand, or a cell from my leg, or a cell from my kidney, every cell have the same genetic constituent. So all the somatic cells in our body should possess the equal genetic information everywhere. That concept should be maintained here. So the first significance of mitosis, which I mean, is equational division. Equational division or equal distribution of chromosomes. I have said before, cell division, there are two types of cell division, mitosis and meiosis. Meiosis cannot achieve this equational division. Only mitosis can achieve this equational division. The second significance of mitosis have surface volume ratio. It's very much required for maintaining surface volume ratio. Volume ratio. If you observe carefully in the beginning of our lecture, I have said the genetic material in interphase itself divides themselves before the cells started dividing. So in the interphase stage, if I have one pen, in one box, it occupies less space. If I have two pen of same size in the box, the content increases, so the size should get increased. That what happens in the cells. When the size of this box increase, the surface also increase, so the surface area increase. This is not a proper judgment to that cell. So what happens? So to reduce the surface volume ratio, the cells divide itself. When the cell divides itself into two cells, instead of a big cell, we have two balls in the same container. The volume is less. That's what I mean to say. Instead of I have a big cell together of same size inside, if I have two daughter cells, the surface is reduced. This what happened here. So cell division is required for reducing surface volume ratio. Hope I made clear this point. The third thing which I mean to say is nucleoplasmic ratio. Nucleoplasmic ratio. So the growth takes place for the entire cell. It contains 6 trillion cells, I have said. So whatever genetic material I have, it should spread to these many cells. So it maintains a nucleoplasmic ratio, an equal nucleoplasmic ratio to be spread to whole cell. I can say I am growing only if I have many number of cells, not I have a big cell. If I have a big cell, that is a disease, right? So I am having 6 trillion cells equally distributed to my entire body, my jumbo body, from a single cell which started as a zygote. This what is called nucleoplasmic ratio, which can be achieved only through mitosis. Then the three things we are aware of it, and the fourth significance of having mitosis is repairing. Here I want to stress one more interesting fact of our body. There are nearly 5 million cells are replaced per day. 5 10 to the power 9 cells are getting repaired and they are getting replaced. Just imagine, for 5 million times a cell is dividing and getting replaced continuously, apart from your normal activities of that liable or these cells, okay, stable cells. Except that permanent cells, liable and stable cells are dividing continuously or discontinuously. So for repair itself, nearly 5 million cells are getting repaired through mitosis. The fifth thing I want to say of significance of mitosis is opportunity for differentiation. If I have more cells, if I have only few cells, there is no possibility of assigning labor to say two cells to different labors. But I have a huge number of cells of equal capability. I can assign these cells to be liver cells. I can assign these cells to blood cells for different properties. So the opportunity for differentiation occurs only through mitosis. So it's very simple and clear, friends. What is mitosis and what is a cell division? What means the flashback of the story, and what is mitosis, and apart from that, what is the significance of mitosis? If you know the significance of mitosis, you can wonder how the cell is greatly doing the mitotic process. Join us for more such videos and make your competitive exam success with Science Teaching Academy. Follow us on Facebook, YouTube channel. Thanks for your subscription. Thanks for your concentration. Thank you.